Hi, good morning, everybody. Good afternoon, good evening, wherever you are. My name is Valerie from the University of Liège. Guest today is Valerie Castellani. Before introducing Valerie, I just would like to inform you that the worldwide neuro organizers have decided to include selected short talks from junior scientists that will be given just before the main presentation. So please feel free to send your CV with a short summary of your research to Denis Jabaudon. This is open to any junior scientist, whatever the country or the research institute, because at the end, only science matters. And yes, there's going to be some training session that will be organized to help the speakers. So now I'm happy to introduce Valerie. So Valerie did her PhD with Jürgen Boltz at the Institut Cerveau et Vision in Paris, where she studied the mechanisms of brain wiring. She next traveled south to France to join the team of Geneviève Rougon, where she did the postdoc at the IBDML Institute in Marseille. And she is now director of research of the CNRS, and she lead a lab at the Neuromyogène Institute in Lyon since 2003. She has a long-lasting interest in axonal guidance mechanisms, using as models the navigation of commissural projections. Well, her team indeed studies the mechanisms by which cells get topographic information to properly orient their polarity processes during the development of the nervous system, using the chicken and mouse embryo as animal models. When she started the lab, she focused first on the analysis of secreted semaphorin family members. Her lab showed that the neuropilin receptors form complexes with IgCAM molecules, which make it possible to integrate regulations of adhesion during growth cone reorientations. She also obtained an ERC consolidator grant to investigate earlier roles of axon guidance signal, and the lab found that these cues provide positional information to spinal cord progenitors in order to orient their division axis. The lab also identified the existence of a molecular memory that allows progenitors of sensory neurons in the dorsal root ganglia to transmit the polarity of shape to the daughter cells. Finally, the team transferred this very interesting question of topography and their experimental approaches to the topic of pediatric cancers, with the objective of addressing tumorigenesis, and metastatic dissemination from the angle of developmental biology, addressing the specific interaction of tumoral cells with their immature microenvironment. And in that context, Valerie co-founded the startup OncoFactory in 2016 to provide, I would say, a la carte patient-derived xenograft cancer models using avian embryos to entangle the pathomechanisms and set up innovative drug screening approaches. She also received the prize Coup d'élan of the Fondation Bettencourt-Schuller in 2017 for her study of neuroblastoma, and she also got the Médaille de l'Innovation from the CNRS in 2018 for translating her basic research in the startup OncoFactory that we just mentioned. So it's my great pleasure now to leave the floor to Valerie and listen to her science.
Okay, thank you very much. There is an echo, I'm sorry for the connection. I hope you can hear me. So thank you very much. Hello, everyone, and thank you, Denis and Laurent, for this presentation. It's my great pleasure to share with you the work of my lab. And I'm going to talk about the mechanisms of axon guidance, which we study for years in the lab, and also about our more recent interest into how these mechanisms could be exploited or possibly hijacked in the context of childhood malignancies. So I'm going to share my screen. I hope you can see my screen. So, because I don't see you, so anyway, Laurent, if something is wrong, you can tell me.
You're all
So
good.
the general,
You're all good.
Okay. So the general question we are interested in the lab is that of how do cells get oriented in the space of the tissue during some fundamental processes of the developing nervous system, for example, when progenitors divide or when newborn neurons acquire some more complex morphology, and also still during cell migration and axon navigation. So we know from years of work in the axon guidance field that cells get positional information from cues expressed in the environment that exert repulsive and attractive effects on axon terminals, the growth cones, or on migrating cell living processes. And we've been showing that these so-called axon guidance molecules bring much earlier contribution to the developing nervous system, providing topographic landmark to dividing progenitors to orient their division axis. But today I'm gonna focus on axon navigation. So our favorite model to ask our questions is the developing spinal cords with commissural axons that navigate the midline in the floor plate, and also associated nerve ganglia that arise from a population of cells that is transient and unique to vertebrates that lie at the dorsal edge of the neural tube, that is the neural crest. And as Laurent said, we got interested in a closely related pathological context, which is the neuroblastoma. That is a cancer that arises from a subpopulation of the neural crest, the one that is committed to build the sympathetic ganglia and the adrenal gland. And these are the sites where primary tumors form in the patient. So it is interesting to consider that both these physiological and pathological processes can be somehow concomitant, which raises, to my point of view, very interesting questions regarding the impact that the unique context of the embryonic development may have on the biology of these tumoral cells. And investigating what share neuroblastoma cells and their cells of origin, the neural crest, should be very helpful to to improve our knowledge of this pathology. And this is what we try to study in the lab by focusing on the process of cell migration and axon guidance genes. So I'm gonna start with the mechanisms of axon guidance. How is encoded the diversity of axon paths that is needed to guide all axons towards the target is still to me a fascinating question, and yet rather not unsolved despite extensive years of work in the field. So obviously we know that a first degree of diversity is conferred by combinatorial expression of receptors that enable subsets of axons navigating in the same environment to make proper guidance decisions. But we know that this is not sufficient for guiding all the axons towards their proper target, and things are much more complex. And we know that in particular, since the remarkable work of Mu-Ming Pu and Kyung-Soo Ong that revealed the potential of the growth cones to vary their sensitivity to the guidance cues, switching from insensitive state to sensitive state, reversing repulsion to attraction, and vice versa. And this, in principle, gives an enormous flexibility to the perception that the growth cones could have of the environmental content, which is exactly what we need, what we want to guide many axons, to generate large numbers of trajectories with a rather limited set of initial guidance molecules. So the challenges are now to understand how and when are these regulations taking place, and what is actually the guidance decision that is triggered by these answers. by these switches of responsiveness. Midline crossing has been established over the years as a great context to study the plasticity of gross-scan responses. I'm going to not to present you the commercial circuits we already had in this cycle of conferences, great talks about commercial circuits. This is just to remind that these are the circuits that interconnect the left and half sides of the central nervous system in bilaterals. They are present in vertebrates, in invertebrates, and throughout the central nervous system. And during their development, all commercial axons face a common task, that is to cross the central midline once and only once in order to project onto contralateral targets. This is a schematic drawing of the navigation of commercial axons in the spinal cord from dorsally located interneurons. They navigate ventrally and they cross the floor plate and they cross the midline within the floor plate prior to turning rostrally to ascend towards higher levels. And in the lab, we focus on the repulsive forces that help the axons navigating the floor plate. So the current view is that the axons are initially insensitive to these local repellents, and they will switch on their sensitivity after midline crossing so that the signals will push the gross cones away from the midline, and they will set a barrier that prevents the axons from crossing back. So many labs, including ours, reported various ligand-receptor pairs that mediate these repulsive forces, essentially slit proteins, slit 1 to 3, that are processed into N-terminal fragments. These are the slit proteins that bind to roundabout receptors, ROBO1 and 2. And we've been showing in the lab that the C-terminal fragments resulting from the processes are indeed also biologically active, and they mediate a guidance effect via another receptor, that is plexin A1. And we also have in the floor plate the semaphorin 3B that exert a guidance effect via a complex of plexin A1 and neuropilin 2. So despite this work, it still remains unclear how do the axons can navigate a repulsive field, with many questions that have been left open, and whose answers could really highlight our understanding of the navigation of the floor plate in particular, and intermediate targets in general. If we focus on the floor plate, how do the ligands, slit ligands, distribute? Do the different ligands play the same role? And actually, how does the physical environment of the axons look like? These are the questions we've been interested in the recent years. And we've been working on the generation of fluorescent tools to report the dynamics of guidance receptors, the ligand patterns, and for live monitoring of the growth cones navigating in their native environment. And I will bring you through the work of 3 great PhD students of the lab, Aurora Pignata, Hugo Ducuin, and Thibaut Gardet. So we first ask by investigating when are guidance receptors available at the growth cone surface, which to us give a rather good idea of when the growth cones can actually perceive the different ligands during the navigation. So to do so, we took advantage of the properties of the pH lurin that is fluorescent at neutral pH, and therefore reports the pool of cell surface proteins. So we fused the pH lurin to the coding sequence of guidance receptors, and we introduced the vectors in the neural tube of the chicken embryo. And then we opened the spinal cord like a book, which is very convenient to looking at axons navigating the floor plate and then turning rostrally, selecting either the ventral funiculus or the lateral funiculus. So this is a kind of movie we can obtain, and you see the axons navigating the floor plate, and the yellow signal indicate that the receptor has been sorted at the cell surface. So we generated cartographies of the special positions when the growth cones turn on the yellow fluorescence. And this way, using this paradigm, this revealed there's a temporal sequence of receptor membrane insertion. So we have first neuropilin 2 that is present from the onset when the axons enter the floor plate. And then we have plexin A1 sorted during the first half of the floor plate navigation, followed by Robo1 in the second part of the floor plate. And then we found Robo2 is never sorted during the floor plate crossing, unlike we thought, but rather much later on the axons that choose to navigate the lateral funiculus and not the ventral funiculus. So this temporal sequence of receptor cell surface sorting provide a basis for the generation of a temporal pattern of repulsive activities during the floor plate navigation, which strongly supports that the different ligands have a very specific outcome. So the next question we ask is, how does the axon navigation path look like? We know a few about this. The morphology of the floor plate glia is thought to be like typical of a progenitor. So floor plate glia have a progenitor-like bipolar morphology with an apical and a basal anchor, and the axons navigate within the basal processes. Close contacts between basal processes and navigating commercial axons were reported in early work based on electron microscopy. So we decided to further investigate the morphology of the floor plate glial cells, and what we did is to perform a sparse electroporation with OXA1 GFP construct that drives the fluorescence within floor plate cells. And then we reconstructed the morphology of individual floor plate glial cells. And as you can see from these images, we were quite surprised to see that these cells do not have such a simple morphology, but they are much more complex. In particular, at the level of the basal processes, there is not a single process, but rather several ramifications, typically 4. And you can see also that they have a particular shape. They are enlarged along the navigation axis, and they are flattened along the rostrocaudal axis. Therefore, they form sort of narrow and aligned corridors. So next, we electroporated MAT1 MB2 MATO construct that drive the fluorescence within commissural axon neurons and the axons, and we could see how the growth cone looks like. And I don't know whether you see the movie. What the movie is supposed to show you is that the growth cones have a particular shape that indicate they adapt their shape to the narrow corridors, like if they were confronted to repetitive contact with the surfaces of the ramified basal processes. So after this, we wondered about the distribution of slit ligands in this mesh of basal processes. We don't know so much about this because using antibodies to detect secreted proteins is very difficult, as you know, and also these antibodies would not distinguish slit N fragments from slit C fragments, and would not also distinguish the full-length proteins from the processed fragments. But from the current model, we imagine that these cues would form sorts of gradients that the growth cones would descend after crossing when they get sensitized to these cues. So to address this question, we took an alternative approach, and we generated a dual-tagged construct flanking the coding sequence of SLIT2, on one side with the cerulean and on the other side with the venous. And then we introduce the vectors in floor plate cells, and so that after endogenous processing of SLIT protein, then the two fluorescents will report individually SLIT C and SLIT N fragments. And this is what we could observe. As you can see, SLIT2N and SLIT2C are detected in the axon navigation path, and clearly they form clusters that have a columnar pattern, indicating that these clusters are deposited on the ramified basal processes of the floor plate glial cells. We quantified the fluorescent signal, and calculating the Pearson coefficient that indicates the degree of fragment segregation. And we found that as measured as we get far from the apical domain, then slit C and slit N fragments become segregated, so that they are segregated in the axon navigation path. So we wondered about the distribution of the full-length protein, and then we substituted in the dual-tact construct SLIT2 sequenced by a non-cleavable mutant. And then what we found is that the signal is no longer detected in the, or very strongly reduced in the axon navigation path, indicating that SLIT2 integral protein is restricted to the apical compartment. And this is also indicated here with this quantification of the basal on apical signal, showing a drop in the mutated condition compared to the wild type for both SLIT2 and N and SLIT2C fragments. So from this data, it appears that SLIT2 proteins is restricted, full-length protein is restricted to the apical compartment of the floor plate glial cell, when where its processing might occur, leading to the generation of slit C and slit N fragments, and then their basal deposition, on the axon navigation path. So this gives a major role to the mechanisms of cleavage of SLIT2, since we need this cleavage for having slit proteins within the navigation path. The mechanisms of cleavage of SLIT2 are largely unknown. There's just a paper that came out last week from Kid Lab showing, reporting that Tolkien protease cleave slit proteins in Drosophila. And there were also a previous report showing that the pheromone convertase amontillado can cleave a slit in the embryonic tissue. In vertebrates, the homologue of amontillado is the pro-protein convertase PC2. So we got interested in looking whether PC2 could cleave vertebrates SLIT2. And this is a Western blot showing the full-length condition with the detection of SLIT2C fragment that report the cleavage. Here is the non-cleavable mutant, and you see that SLIT2C fragment is gone. And when we expose the cells with CMK, that inhibit PC2, then you can see that there is a strong reduction of SLIT2C signal, indicating that this inhibitor, that PC2 is able to cleave vertebrate slit. So interestingly, we also found that PC2 is expressed at the right time and position. It is detected in the floor plate in chicken embryos, as you can see from this immunolabeling. And thus what we did is to combine our expression of dual-tact SLIT2 with a pharmacological injection of the inhibitor to chicken embryos at 2 time points. And then we quantified again SLIT2C and SLIT2N fragments, and we could observe there is a reduction of the basal on apical ratio, indicating that inhibiting PC2 resulting in a reduction of fragment deposition in the axon navigation path. So then the next question we wanted to address is what's the mode of action of these slit fragments that are deposited in the basal processes. And we focus on slit C plexin A1 signaling, which we found in our previous work. And we worked on the side of the receptor by generating a knock-in mouse model in which plexin A1 carry a particular mutation on a tyrosine, which we found in this previous work to alter slit C signaling, whereas we found it preserved the responses of the growth cones to SEMA3B. We analyzed this mouse model in light sheet microscopy using Robo3 as a commercial marker. And you can see that there is a disorganization of the navigation of the floor plate. We quantified the phenotype in this mouse line using dye tracing of open book preparation. So this is the normal path. And what we observed is the presence of recrossing phenotypes that are quantified here. We found recrossing both in heterozygous and homozygous embryos that we never see in the wild-type littermates. And this recrossing phenotype is reminiscent, very typical of the triple deletion of CYP123 proteins, as well as the deletion of plexin A1. Whereas we don't see any stalling. The stalling is when the axons are arrested within the floor plate. And this stalling phenotype is typical of the deletion of SEMA3B and also plexin A1. So this means that specifically releasing CT signaling induces recrossing and trajectory disorganization. Next, we expressed the pH lurine versions of wild-type and mutated receptors back to plexin A1 knockout embryos. And in living open book preparation, we could observe that the growth cones that sorted the wild-type receptor were capable of normally navigating the floor plate, having a straight growth. Whereas in contrast, those growth cones that sorted the mutated receptor started to deviate their trajectory and also to turn back on themselves. And this is also seen with this qualitative index from 0 straight growth to
Valerie, I think you are freezing, at least on my screen. So maybe you should refresh just to make sure that you can pursue.
yes.
Can you hear me?
I'm
Yeah, so you have to restart. Yeah. Yeah.
again.
Yeah, you have to share again. Ah.
It's okay?
Now I can hear you again. So maybe you should try to share.
Yeah.
And go back to the slide. Yeah, that one actually. That's correct.
It's okay?
Yes, thank you.
Okay, so I was saying that we did these experiments also in the chicken embryo, and we found similar results. The growth cones that failed to undergo a straight and forward directed trajectories, and this is a nice example of growth cones that turn on itself. So it was clear from video time-lapse movies that the growth cones that express the mutated receptor undergo a very exploratory and abnormal, highly exploratory and abnormal behavior, exploring locally the environment, turning, so which can lead to deviate the trajectories. So releasing CTC-mediated constraints result in high growth cone exploration. So to sum up what I have shown you, we do not believe anymore, at least for CTC, that the axons acquire sensitivity to these cues only after midline crossing, and that these cues form gradients that the growth can descend. Rather, from our data, what we think is that the growth cones perceive CTC from from the entry. CT is active already during the first half part of the floor plate navigation, and actually CT is deposited on the basal processes, and then through repetitive contact between these surfaces and the growth cones, it forces the axons, the growth cones, to keep a straight growth by preventing any attempt of growth cone exploration that would lead to deviation, which is, to my point of view, a very efficient mechanism to ensure completion of the floor plate crossing, and in particular by counteracting any deviation that would occur if the growth cones already perceive the longitudinal gradients of guidance cues that have to trigger the turning of the growth cones only after they have exited the floor plate. So this might also be a general mechanism for navigation at intermediate targets. So I'm going to leave you with this model and switch to the dark side of axon guidance genes, from neurodevelopment to pediatric cancer. So this is a work that we conduct with Céline Deslois-Bourgeois, who is a senior scientist in the lab, and she joined the lab several years ago now as a postdoc. And our aim is to study pediatric cancers under the angle of the developmental biology, considering that pediatric cancers are far different from adult cancers in that they develop in still immature organisms. And this can occur very early. So at a time when morphogenetic programs are highly active, supporting migration and proliferation to build tissues and organs. So the idea was to transfer the experimental approaches of the developmental biology to pediatric cancers, but also to study developmental signaling that cancer cells could take advantage of or possibly hijack. So as I said in my introduction, we got interested in neuroblastoma. This is a cancer with an embryonic origin. It arises from the sympathoadrenal neural crest. This cancer is the most frequent extracranial solid tumor of children below 5 years of age, and actually half of the patients are below 2 years. These are the primary tumor sites, and aggressive forms are devastating and highly metastatic, with multiple foci in the lymph nodes, the skin, the liver, the bones, and the bone marrow. In vivo models lack to study the biology of neuroblastoma, in particular models that would recapitulate the embryonic context. And this is a reason why we decided to set up a novel model to study neuroblastoma, taking advantage of the avian embryo, which we knew from years of work from the developmental biology, and especially the work of Nicole Ledouarin, that it was very suited to chimeric contexts. So we started with human neuroblastoma cells that we transduced with fluorescent reporters. And then what we did is to implant the cells back to their sites of origin, the neural crest, with the naive idea that maybe these cells could replay the history of the disease. And we were quite struck to see that these neuroblastoma cells, human neuroblastoma cells, were fully competent of migrating ventrally, recognizing the sympathoadrenal territories of the embryo, where they arrested exactly as their physiological counterparts, the neural crest, and but in which they could form huge tumors. And not only they recapitulate the primary tumors, but also we observed at late stage that some cells detach from the primary tumors and engage towards dissemination, and we could identify the dorsal aorta and the peripheral nerves as the main migration paths of these cells. So having this model in hand, it was possible to ask a series of questions, in particular trying to understand what neuroblastoma cells and neural crest cells share. And the first question we ask is whether neuroblastoma cells could retain the typical mode of migration of the neural crest that, as you probably know, undergo a very efficient collective migration. So we set up live imaging of thick slices of grafted embryos, and you can see from these movies that neuroblastoma cells that are migrating in the embryonic tissue to reach the sympathoadrenal territories migrate collectively, either in mass, as here, or they could form migrating chains. We then wanted to identify or to investigate the gene program that could prepare neuroblastoma cells to metastasize in the primary tumors. So we dissected out the tumors in the primary sites, and we conducted a transcriptomic analysis comparing the transcriptome in the primary tumors to the transcriptome of the same cells before the grafting. And using in combination with functional studies, we could actually show that neuroblastoma cells keep active a cell-cell cohesion program during that collective migration and during and within the primary tumors that maintain the collectiveness of cells together, and that at some point some cells shut down this cell-cell cohesion program and start detaching from the primary tumors and engaging into dissemination. And here comes the axon guidance genes in the scheme, because we found that at the core of this cell-cell cohesion program is the semaphorin, plexin, neuropilin, and signaling pathways. So to make it very short and with a focus on SEMA3C, we could observe that knocking down by reducing by twofold the levels of SEMA3C in neuroblastoma cells prior to the grafting totally disrupted. You hear me? Is there a problem?
No, it's okay. I mean,
Oh,
all good
there were
so far.
Okay. So what happens when we knock down SEMA3C and we reduce by twofold its level within neuroblastoma cells is that these completely disrupt their collective migration. They are no longer capable of forming primary tumors, but rather they disseminate throughout the embryonic tissues, meaning that high SEMA3C levels is required for this collective migration. And this is even clear if we make these experiments by grafting in two colors, the control cells and the knockdown cells, and you see that the control cells perfectly form tumors, whereas the knockdown cells, they disseminate. And we found similar results for neuropilin 2, 1, and the plexins A2 and A4. So now if we knock down SEMA3C levels later on when cells already form primary tumors using an inducible tool, what happens is that we boost the metastatic potential of these cells, and we can rescue back the metastatic profile back to the control, and even better if we re-express SEMA3C. So this is an example of sharing of a signaling pathway, the semaphorin plexin neuropilin pathway, by neuroblastoma cells and neural crest cells, since the semaphorin signaling has been well shown to guide the migration of the neural crest, and in particular for SEMA3C, the cardiac neural crest cells. So from this initial finding, we've been working in three directions, and I'm going to give you an overview of where we are, setting light microscopy and live imaging to further look at the behavior of neuroblastoma cells in our model, working towards the identification of microenvironment-specific gene signatures of neuroblastoma disease progression. And we also work on searching for sympathetic-derived signals that could regulate the cell-cell cohesion program and trigger the metastatic onset. So this is an image of light microscopy. We are 2 days after the grafting, and the embryo is labeled with an axonal marker to highlight the nerves, and we see the primary tumor formation. Here we are 10 days after the grafting, and we see there's a huge dissemination of neuroblastoma cells along the nerves that convey the cells to the chest. With this live imaging of thick embryo slices, we could also observe the migration of neuroblastoma cells within the dorsal aorta. I don't know whether you can see this. So we see the cells that approach the dorsal aorta, and at some point these cells will change their shape. They become somehow fusiform, they extend sort of leading process, and they enter the wall of the vessel. So now we are 12 days after the grafting, and we see the cells that have reached the skeletal elements, the bone. They are in the bones. And as you can see from this little movie here, they form a secondary foci in the bone marrow. So we have now the full sequence of the disease from the primary tumor to the bone marrow metastases, which are the main metastatic foci in the patients. If we look at a single embryo, then we will get a picture of cells at different stages of the disease. We have the cells that are still, that remain in the primary tumors. We have the cells that migrate along the nerves. We have the cells within the dorsal aorta, and we have the cells in the final location in the bone marrow. So with batches of embryos, we could microdissect the different tissues, embryo per embryo, for sorting individually the cells for either single cell RNA-seq or a bulk RNA-seq analysis. And these are ongoing analyses, but we already see that within the neuroblastoma population, there is 3 distinct cell profiles. And we also already know that all 3 clusters are able, fully competent for migrating along peripheral nerves and dorsal aorta. And all 3 can also reestablish secondary tumors in the bone marrow. And we also know from this analysis that cells that use as a migration path the dorsal aorta exclusively or almost exclusively come from the adrenal primary tumor, whereas the cells that migrate along nerves come either both from the sympathetic and the adrenal primary tumors. And we also know that both migration paths can convey the cells to the bone marrow. So we have now plenty of data whose analysis should reveal or should highlight the specific molecular dialogues that these neuroblastoma cells engage with the different embryonic environments they encounter during disease progression. So in the last couple of slides, I will show you the work of another PhD student of the lab, Dunya. She was interested in searching whether signals coming from the sympathetic environment could influence neuroblastoma cells and trigger the metastatic behavior. So to do these experiments, we use a cell-cell cohesion assay that was set up in our previous work, which consists in forcing neuroblastoma cells to form a dense mass that resemble a primary tumor. And then we can play with a gene-gene cohesion program, knocking down genes or putting what we want in the medium so that we can try to see whether we can disaggregate the cells. So what we did is to micro-dissecting out the sympathetic chain of chicken embryos at different time points, and then we culture the sympathetic chain to produce conditioned medium, and we expose the tumoral masses to this conditioned medium. And as you can see from this analysis, we found that the sympathetic conditioned medium was fully able to trigger the disaggregation of the tumoral masses. And this effect decreased over time. We repeated the experiments using a second neuroblastoma cells. And we also did this experiment using a sympathetic chain dissected from mouse embryo. And as you can see, we found similar effects. So the sympathetic ganglia release signals that are able to induce this disaggregation, this cell disaggregation in the mass. So to analyze, to identify the active signals, we went to mass spectrometry analysis in collaboration with Johan Coutet from the CEA of Grenoble. And we analyzed different conditioned medium conditioned with sympathetic ganglia, but also dorsal ganglia, which we found was also efficient to trigger the disaggregation of the tumoral mass. And by crossing all these different analyses, we went with a list of proteins that have very interesting reported functions regulating migration and adhesion, which are the kind of processes we are looking at. In parallel, we worked on the sign of neuroblastoma cells, and we analyzed the transcriptome of cells in the tumoral masses and after exposure to the conditioned medium. And we compare the regulated genes with signatures of cell-cell contacts, cell matrix contacts, cell migration, and we could generate a sympathetic-induced neuroblastoma cell detachment-derived signature, which we were very happy to see that it contains the semaphorin plex in European signaling, which we found in our previous work through different approaches. So to select a novel candidate signaling for further functional studies, we cross all our large-scale molecular analysis, and we choose to work with the olfactomedin pathway. Olfactomedin share with other proteins, olfactomedin domains. We have, for example, the laterophilins that were reported in a great work from Elena Seradaki and Rüdiger Klein laboratory to act to mediate repulsive guidance via tenorin in the context of cortical cell migration. Olfactomedin 1 protein has a very interesting expression profile, as you can see from this picture. It is expressed in the developing peripheral ganglia, and it has already been implicated in the development of the neural crest. This is just a schematic drawing of the binding partners and signaling effectors of olfactomedin proteins. And this is just to show you that in our initial transcriptomic analysis, we found several members that are regulated. And also in our experiments with the sympathetic conditioned medium, we also found a regulation of several members. So to address the function, the property of olfactomedin protein, we expose tumoral masses to recombinant olfactomedin protein in different doses, and we found there is an effect of this protein at these doses. So application of olfactomedin 1 can mimic the sympathetic conditioned media. And if we incubate the conditioned media with a function-blocking antibody to olfactomedin 1, then the disaggregation went back to the control level. So we rescue the cohesion of the mass back to the control level. So what we did next is to inject the anti-olfactomedin antibody to grafted embryo, and we analyzed in microscopy the metastatic process. And these are two representative pictures and the corresponding movie. And you see that the spread of the metastasis is more important in the control condition than in the treated condition. And we found there is a significant reduction in the number of metastatic foci. And to further quantify this experiment, we color-coded the metastasis and relative to quantify the distance of the foci to the primary tumor sites. And this way we could observe that in the antibody-treated condition, cells migrated at less distance than in the control condition, which is indicated here. But in contrast, we found that cells migrating less, but they form tumors that had a higher volume, indicating that there is dissemination and proliferation balance. And actually this makes sense if we consider that blocking olfactomedin 1 then will keep the cells together, which is an important condition, as we can see from the primary tumor, for their proliferation. So to sum up what I have shown you, we've found that neuroblastoma cells actually take advantage of signals that are released by the sympathetic ganglia during their physiological development. and they use these signals to disseminate. And we identify among the signals olfactomedian protein. So beyond this data, I think this study illustrates to which extent the unique context of the embryonic development can provide opportunities to cells that undergo malignant transformation. And these malignant cells use this opportunity for widely disseminating. And it can explain, at least in part, why aggressive forms of childhood malignancies are so highly metastatic. So I believe that also that better characterizing the mechanisms of action of axon guidance genes, both in the physiological and the pathological context, should open new ways for treatments for the patients for these aggressive forms that remain incurable. So I would like to thank all my great team, all people that contributed to this work, and also our clinical and academic collaborations. And this is our support, and I thank you very much, and I can take your questions now.
Thanks, Valerie. So you can unshare your screen now.
Alors, how to do that?
Oh. Okay, perfect. So thank you very much for this very comprehensive presentation. I learned a lot. So we have a couple of questions that I will basically read and leave you time to answer. Yeah, please, guys, if you have any comments or questions, just feel free to drop it in the Ask a Question box at the bottom of the screen, like I mentioned before. So let's start. Okay, so how do neuroblastoma cells enter vessels? What are the mechanisms used for these cells to enter the dorsal aorta, for instance?
Yeah, this we don't know. We are really at the beginning, and we really expect to get some insights from our molecular analysis. And this is actually the reason why we went through all this molecular analysis to try to pick up some signalings, and then we can conduct functional studies. So we don't know.
And in your xenograft model, is it also happening? Yeah, actually, it is happening in the xenograft model, right? In the chicken.
Yes. Yes. Yes.
So do we know whether this is also happening in mouse model? If there's any mouse model that could be used to look at it?
Actually, no, that's the problem. And the problem with mouse models is that neuroblastoma cells are grafted in adult mice. Obviously, in some cases, people graft them in the sympathoadrenal tissues, but this is an adult context. And when cells are grafted, as usually under the skin, there is no metastasis at all, so it's very difficult to study this process, and this is why with this model we can now have a way to address these questions.
Yeah. Okay, so the next one is about the first part. It's me also asking, sorry. So you show these beautiful slit C mechanisms for four-plate crossing regulation. And I was wondering whether this is conserved throughout the neural tube, if you look antero-posteriorly. Because it was shown at a specific place, right? So I was wondering whether this is conserved through the axis.
If I understand correctly, the question is whether the expression or the mechanisms of SIT action is restricted to some rostrocaudal levels. There is no reason of thinking that there is some differences along the anterior-posterior axis. So throughout the spinal cord, we believe this is happening this way.
There's no differences in the kinetics of floor plate crossing. I'm not an expert in that.
Oh, there is a maturation gradient. So obviously, this has to be timed with the generation of the neurons, the migration of the axons, and so on.
So you have you have a gradient, an anteroposterior gradient, or retrocaudal gradient as well?
There are gradients of maturations, and if we look at the expression profile of the ligands, it's not that they are not so much regulated. They are present from E9 to much later than the floor plate navigation.
Then you expect to see similar kind of gradient of these mechanisms, basically, I guess.
The idea is that each growth cone will have its own perception of the environment. It's not so much that the environment will change, actually. It's much more that it's the way the growth cone will perceive the environment that matter for the navigation. And actually, this is efficient since the axons arrive by wave. There are waves of axons.
Right.
And the process of floor plate navigation takes several days, or at least 2 days in the mouse. So you can get the regulation, or you can get the process taking place within each growth cone independently of the expression or changes of expression of the ligands.
Let me jump now on a question from Sergi Puigros. So, well, he said, thanks for the great talk, Valerie. Is it known whether the motor neurons in the spinal cord also release cleaved version of slits?
So it's known that motor neurons express slits. So there are several work that have shown that. And actually, the slit robo pathway is very important for the maintenance of the fasciculation of motor axons together. There are works on phrenic axons showing that from Marc Tessilevin lab, and we have been working also on this signaling on phrenic motor neurons. So whether, there have been not so much interest on SLIT-C proteins until we found it is active. So there is no reason thinking that motor neurons do not process SLIT proteins, and so we think that SLIT-C is also present and expressed by or released by motor neurons, but we did not check by
right. Okay. Yeah. Regarding the last part, which I found very interesting, the proteomics, right? You catch this olfactomedia, right? So I was wondering whether this pathway, noeline olfactomedin, acts for in a cell autonomous or non-cell autonomous way on those migrating cells, or both maybe.
So we don't know really, but in principle they can act both cell autonomously and non-cell autonomously. So from our experiment with our ex vivo experiment, we know that putting the ligand in the culture medium of the aggregate can have an effect. So clearly, this is a non-cell autonomous effect. But we also believe that maybe this is more complex. In particular, there is cross-regulation across olfactomedin proteins, and then we could have an interplay between olfactomedin protein expressed by neuroblastoma cells, and so both cell autonomous and non-cell autonomous effects. So we don't know. But here in our experiment, clearly it's a non-cell autonomous effect.
Okay. Something which is not directly related. I was wondering, looking at your talk and all those mechanisms and pathways, I was wondering whether some of them, if you think that some of them could be used in other cancer contexts, basically. I'm just asking this because I just said this. I mean, there was this publication about slit use for lung cancer, I think. So reusing what we call neuro mechanisms for other cancer, for spreading other cancer. So you believe that this pathway that you are describing might also be
yes, for sure. There are examples, and more generally, all the pathways that are acting during development to guide cells. Actually, they are implicated in cancer, and but it's hard to say. What is interesting is not only to consider these axon guidance genes as genes that can regulate cancer cell in the adult context, because we know this signaling regulates cell motility in general. So they regulate the cytoskeleton. So obviously they can have a plethora of effects. What I think is very interesting is trying to understand the way they play their role during the development, to which extent this make pediatric cancers very different from adult cancers, and so really to understand whether cells that become malignant in an embryonic organism, these are cells of the development. Sure. So these are cells that might be imprinted for having also a particular developmental stage. That is not what happens in adult cancer. Yeah. So because these cues are also continue to be expressed in the adult tissues, it's hard to say whether the role that we play in adult tissues and thus the deregulation in adult cancers reflect actually what happens in cancers with embryonic origin.
So I have a question from a more aficionado of cell migration. So what do we know about how those different molecules act on the remodeling or the dynamicity of the cytoskeleton? Is it something known?
Yeah, plenty of things are unknown, and I'm not a specialist. So for the collective migration, what is very nice, and what is I think is nice in the system is with our findings with SEMA3C and neuroblastoma cells, in that it gives the idea that actually cancer cells communicate together. And it's not like they are free of all what they see in the environment.
Physical link, right?
They physically and they physically interact and they share signals. And I did not show that, but we did plenty of experiments by combining, mixing cells that express different levels of SEMA3C. And we see that we can organize and we can segregate the cell populations according to the levels of signals. So what is known in the collective migration is actually that cells release signal for the collectivity of cells so that they are kept together. So they are attractive signals that maintain the cells together so that they are not lost in the environment. And there are also some cell contact mechanisms. So
you presented like two different set of neuroblastoma cells, right? That were invading different places, if I remember well. So I was wondering, to what extent collective means, if you mix these cells, would they migrate in a collective way, or they really talk only to their population? Is what I mean.
So what is striking with our experiment in using the chicken model is that when we put back the cells in the dorsal edge of the neural tube, then the cells behave, as far as we can observe it, they behave exactly as neural crest cells. They migrate collectively, they ignore vessels, they ignore the surrounding axons, they just migrate collectively towards the sympathoadrenal territories. And then they switch on their tumoral potential. And when they migrate out, so when they engage in secondary dissemination, they have a completely different mode of migration. Right. They migrate much more individually. They directly go to the dorsal aorta, whereas before reaching the sympathetic ganglia, they ignore the dorsal aorta, which was really close, exactly at the same distance. Mm-hmm. So it's like when they escape from the primary tumors, they have reset their migration mode and they migrate now completely differently. And this is with this mode of migration that they migrate along nerves and they migrate within the aorta.
And you believe that these cells that are disseminating from the primary tumors are comparable to the one initially in the tumor? Is it like a kind of fate specification? I mean, does the cell are comparable, the one that are migrated far away from the primary tumor and the one that are staying close together? So what happened basically? Is there any mutation or what triggers this?
We don't think it's a question of mutation. We think this is a question of adaptation to different environment. And so this is what we're going to show with our RNA-seq analysis. And what we already show is that it's not like the cells are different. They are not different. Actually, the diversity of cell types we see in the primary tumors is transferred during the migration, and we find it again in the secondary tumors. But what will what will be different is the set of genes that they activate to communicate with a different environment. And so when they migrate along nerves, I guess they engage some molecular interactions with the nerves. When they migrate within the aorta, this will be a different type of dialogue. And when they reform tumors in the bone marrow, we obviously, and actually what we think here in this case is that their cell-cell cohesion program is now upregulated so that they can form again a cohesive mass, which is highly supportive for their proliferation. So it's this adaptation to the different environments that I think we will be able to really characterize with this approach.
So playing with those signaling, and you were mentioning this at the end of your talk, may serve for future therapy, right? So I was just wondering what we know about the current therapy for this child that is suffering from this devastating cancer. So what
there are not so much current therapies as it's the case for all pediatric cancers, because they receive therapies that have been developed for adult cancers.
Yeah.
So for neuroblastoma, there are some forms that are a very good prognosis. So patients receive chemotherapies and classical treatments. Obviously. As for other pediatric cancers, the treatments are very heavy, so secondary effects can be really huge, which really highlight the need of having therapies that have been specifically developed for pediatric cancers. So there are some, and for metastatic neuroblastoma. These are quite uncurable. They are the two major problems: metastasis and also resistance to treatment. So there are some clinical trials with targeted therapies, with immunotherapy, but obviously not so much.
So far. Yeah. Okay, thank you very much again. I think there's no more question, unless someone wants to drop a last question. But yeah, yes, Valerie.
Yes.
You wanted to say something? Sorry.
No. I just want to say thank you. It was great to discuss with you, and thank you for your question. So,
no, my pleasure. Thank you very much. I just want to make a last announcement before closing the session. So next week, the speaker will be Victor Borrell from Alicante. He will talk about the genetic evolution of the cerebral cortex size determinants. So stay tuned. Thank you very much. Bye-bye.
Thank you. Bye.